Hello class, I'm Perla.
I'm Shanice.
I'm Ricardo.
We will be demonstrating the Kirby-Bauer technique for antibiotic diffusion. This assay is used to test if a bacterium is resistant or sensitive to a substance. The main purpose of this assay is ultimately to see how much of an agent is needed to inhibit bacterial growth. For the purpose of this video, we will only be demonstrating how to use this assay to test if E. coli is inhibited by cinnamon, boric acid, or toothpaste. Then we will be looking at previous plates that we have already prepared to see how different organisms have reacted to these three substances. You will need the following materials seen here to conduct the Kirby-Bauer assay. We will label the outside rim of the Petri dish on the agar side. Always label with the organism name, the treatment, the date, the type of agar medium being used, and the optimal incubation temperature of the organism you want to grow. With a marker, we will also divide the plate into 4 quadrants that correspond to the discs we will place. The first quadrant will be the control, the second the toothpaste, the third the boric acid, and the fourth will be the cinnamon. We will now be preparing the extracts. We will be taking 0.3 grams of each substance and adding it to 1 milliliter of water in a microcentrifuge tube that we pre-labeled with their corresponding substance. Here we are just demonstrating how the substances are measured out. Once prepared, the extracts are shaken for 10 minutes without stopping.
Next, we are going to use a sterile swab to inoculate the plate using the same method. Grab the test tube and remove the cap and gently flame to sterilize. Dip the swab into the bacterial broth. Remove the tube and gently flame sterilize. Then recap the tube. Next, pick up the plate and using the swab in a side-to-side motion, swab the plate gently. The idea is to create a uniform layer of bacteria. To do this, slightly rotate the plate. When the entire surface of the plate has been covered, dispose of the swab in a biohazard container. Flame sterilize the forceps and make certain the ethanol has burned off completely and allowed to briefly cool.
Pick up a disc carefully, dip it halfway in the treatment created, and place it on one quadrant of the plate. Tap lightly on the disc to ensure it stays in place. Remember to use a flame to sterilize the forceps between each disc placement. After all discs have been placed, the plates will be placed into the refrigerator with the agar side facing down for around 10 minutes. This will help delay growth of bacteria while the substances diffuse into the agar. Then the plates are parafilmed shut and placed into the 37-degree incubator and left to incubate for 24 to 48 hours. Plates will be inverted as usual when being placed in the incubator with the lid facing downward. After incubation, bacterial growth should be seen as colonies. If no growth is seen, or if the colonies are too weak, then it is either due to human error or the wrong medium being used.
Before discussing the results, we would like to go a little deeper into our scientific reasoning. As you may or may not know, E. coli is a very common bacteria that lives inside the human body and is a pathogen that can cause a lot of infections. We chose to test how well toothpaste inhibited the growth of E. coli because toothpaste is supposed to protect the oral cavity from bacteria. A study investigated this further by looking at how different toothpaste formulations inhibited the growth of E. coli. The following ingredients found in many toothpastes significantly inhibited E. coli growth.
As for the cinnamon, studies have shown that it is effective in preventing the spoilage of food because of its antimicrobial properties. However, this other study has shown that bacteria like Bacillus megatherium are not affected by cinnamon. Bacillus megatherium is a non-pathogenic soil bacteria that actually aids in a wastewater treatment by remediation and biodegradation, cleaning and deodorizing, drain and septic treatment as well as enzyme and chemical production. When we discuss the results of past disc diffusion assays, we will see that the Bacillus megatherium reacted to cinnamon in a very interesting way.
Now moving on to boric acid. Boric acid has been used in the past to treat bacterial and yeast infections due to how toxic it can be to microorganisms. One of these microorganisms is Serratia marcescens. This bacteria is a pathogen that is responsible for a lot of hospital-acquired infections. This study investigated how effective multi-purpose disinfecting contact lens solutions were at inhibiting Serratia marcescens growth. The results showed that contact solutions that contained boric acid were cytotoxic to this bacteria. Now we will be looking at previous plates that have been tested. This plate shows the effect toothpaste has on E. coli. Notice the zone of inhibition around the disc containing toothpaste. The diameter of the inhibition zone is a measure of sensitivity to a compound. The greater the sensitivity, the larger the zones will be. As you can see, there is a 35mm zone of inhibition for toothpaste. This would suggest that E. coli is highly sensitive to toothpaste as we had expected. In this next plate, we have a culture of Bacillus megaterium. Interesting enough, instead of a zone of inhibition, we have some swarming behavior around the disc with cinnamon. This area measures around 25mm. In this next plate, we have a culture of Serratia marcescens, with the third quadrant testing for boric acid inhibition. There is a 15mm zone of inhibition, so that means that Serratia marcescens is sensitive to boric acid, as expected.
An unexpected result was definitely the swarming behavior of the Bacillus megaterium that was exposed to the cinnamon. Swarming behaviors usually occur when the bacteria is supplied with a nutrient they really like. In summary, the Kirby-Bauer assay is a very effective way of testing the inhibitory response of the organism you are trying to grow. The diameter of the zone of inhibition can give us scientists an idea of how sensitive the bacteria are by inhibiting growth and not directly cleaning the bacteria.
So we hope that this video was helpful and served as a good refresher. Thanks for watching. Bye!